Hello, welcome to our webinar. My name is May. I'm the Business Development Director for TriLink. Prior to TriLink, I worked for Promega, Agilent, and Syndigo. I have over 15 years of experience in drug discovery and molecular biology. My goal here is to help customers with their mRNA projects. I certainly like to see more mRNA used in both research and therapeutics. When people talk about mRNA, lots of times the focus is on the coding sequence, but there are a lot more to mRNA than the coding sequence. There is a 5' UTR, 3' UTR, cap, and tail. In the cytosol, the cap structure binds to the initiation factor EIF4E. The tail also binds to the initiation factor EIF4G. Those binding bring the mRNA into this loop structure. And this structure really helped to stabilize mRNA. We've known mRNA for a long time. But for a long time, people thought it was not possible to use mRNA as a therapeutic because it's not stable. So what has changed? In our mind, there are two big breakthroughs. One is modified uridine. This one is N1-methylpseudouridine. This is in the Moderna vaccine and also in the BioNTech vaccine. The other big contribution is this cap1 structure. If you look at this cap, the first nucleotide on the 2' O position, there's a methyl group. So this is a cap1 structure, and you can achieve this cap1 structure with our CleanCap technology. How do those two technologies help to stabilize mRNA? Because both help the mRNA to evade the immune response. This is your mRNA in an LNP. When it comes into cells, it merges with endosome. In endosome, there are TLR receptors, TLR7A, TLR3, recognize double-strand RNA or single-strand RNA. In the cytosol, you have RIG-I, MDA5. Those also recognize double-strand RNA, and specifically, they also recognize improperly capped mRNA. For example, a cap0 structure has higher binding to RIG-I and MDA5. As a result of the signal transduction, it turns on genes in the nuclease. If this happens in vivo, it triggers pro-inflammatory cytokine response. If this happened in a petri dish, it means cell can go to apoptosis. Let's talk about modified uridine first. Those two are seminal papers from Dr. Carrico and Dr. Weissman. I listened to a talk by Dr. Carrico. She talked about she really wants to make mRNA into a drug. And when she transfected mRNA and transfer RNA into cells, she discovered the cell died after taking in the mRNA. However, transfer RNA did not kill the cells. And what's the difference? She discovered transfer RNA had a high percentage of pseudouridine. Pseudouridine is actually naturally occurring isoform of nucleoside. And so that's this paper. She discovered the pseudouridine or modified uridine can really help the mRNA to evade immune response. This is a more recent paper from Moderna. They discovered that N1-methylpseudouridine not only can help the mRNA get around the immune response, it can also improve the ribosome density, meaning improve the protein translation. Those are our data. The gray bar is N1-methylpseudouridine, the blue one is 5-methoxyuridine, and black one is pseudouridine. Across all cell lines, the modified uridines give you much higher protein response in terms of fluorescence signal. And we do sell those compounds. Those are the catalog numbers. What is a cap? The cap is this structure. when we talk about DNA and RNA, we're talking about 5' end to 3' end. So this is a 5' end, this is 3' end. This is a head, this is tail. So the DNA RNA chain is head to tail connection, right? The cap is actually 5 to 5, so it's a head to head connection. So you have two tails, one here and one here. And you also need a methyl group at this position. Once you have the inverted G with a methyl group, that is a cap 0 structure. If you change the OH group into methyl group here, it gives you the cap 1 structure. As we talked about before, cap 1 structure really helped the mRNA to evade the innate immune response, and the higher you chloride, the higher level of methylation, or the higher percentage of the cap 1 structure. This is a comparison of mRNA either with cap 1 or cap 0 structure. Those are firefly luciferase with a different cap structure. We checked the luciferase signal after 24 hours. As you can see, cap 1 mRNA give you pretty good light signal. However, if you use cap 0 structure, you got close to nothing. How the cell add the cap structure? It occurred naturally in the nuclease after the nascent mRNA grew to about 25 to 30 nucleotides. There are 4 steps. This is mRNA. The first step is to get rid of 1P to give you this structure. The second step is to add the inverted G. The third step is to add the methyl group to this position to give you cap 0 structure. If you want cap 1, there's additional methylation step to give you cap 1. So this is naturally occurring in the cells. You can also do it in a tube. However, it's an extra step. So people ask, can we do it co-transcriptionally? The answer is yes, you can add cap analog. When I first came to the mRNA field, my question was, if you add this cap analog, how do you prevent that from getting incorporated into the mRNA chain? The answer is triphosphate, PPP. You really need a triphosphate to get a nucleotide added to the chain. So if you look at the cap analog, the PPP got buried inside. So this cap analog can only be incorporated in the very first position of the mRNA chain. And the first cap analog is ARCA, anti-reverse cap analog. We'll talk about this more on the next slide. This is the first analog. It has its problems. So first problem is low capping efficiency. Still, you get cap 0 structure, not cap 1. So let's talk about ARCA. So this is ARCA. First of all, it's a dimer. There are 2 nucleotides, identical. This is inverted G, this is G. And on this 3' position, you have a methyl group. Without the methyl group, you can have nucleotide incorporated here. So once you have that added, the nucleotide can only be added here. So that's why it's called ARCA, anti-reverse cap analog. When you use ARCA, this is ARCA, anew in the first position of your DNA template, and this is GTP, G triphosphate. Those two compete with each other. In order to drive the reaction this way toward ARCA, you have to increase the ratio of cap to GTP. In our hand, we actually use 1:4 ratio GTP versus cap. Even if you increase this ratio, the capping efficiency is still low. It's only 70%. And it also caused some problem because you essentially have to reduce the level of GTP. By doing that, you sacrifice the yield. So the yield of this capping analog is kind of low. The other problem is if 70% of the mRNA is capped, that means 30% is actually G triphosphate. That is pretty bad because 5' and free hunting triphosphate is very immunogenic. So if you use ARCA, you have to use phosphatase to get rid of the PPP at 5' end. The bigger problem of ARCA is it's still a cap 0 structure. There's no methyl group here. I talked to my scientist and I said, well, if you can add a methyl group here, you can add methyl group here. Problem solved. Not so simple. If you add this dimer with the methyl group here into the IVT reaction, T7 polymerase will have a hard time incorporating a nucleotide here, if here is a methyl group. So we solved that problem with our clean cap. So here is a clean cap. First of all, it gives you cap 1 structure, right? Here's the methyl group. In order for T7 polymerase to continue to add a nucleotide, we add another base behind it. So here, if you look at it's OH and OH, and T7 can continue to elongate the RNA chain. First of all, our clean cap is a trimer to give you a cap 1 structure. There's 3 bases, right? Our first generation of clean cap is clean cap GG. However, GG still compete with GTP, and your capping efficiency still hover around 70%. We solved that problem with AG. So for the first base, instead of the G, we add A here. Turns out T7 polymerase does not like A. You can't add A by itself. So by having an AG clean cap, we can have a very high capping efficiency. We routinely get 98% capping efficiency, and we tell people we guarantee 94%. Because with AG, you don't have to compete with GTP, so ratio can be reduced. Remember, ARCA is 1:4, right? Here we use 5:4 ratio. We actually use less cap analog than GTP. So that really helps to drive out the yield. So in this case, we get 2.5-fold of yield, mRNA yield, compared to ARCA. Those are commercially available clean caps. One is clean cap AG, just simple AG. And this is a clean cap AG with 3' OMEs. This is kind of like ARCA version of clean cap. We have another clean cap AU. We designed that for self-amplifying mRNA. What's the difference? So instead of having mRNA here, we add RNA replication machinery, non-structural protein 1-4 from alpha virus. Turns out this replication machinery starts with AUG. So if you use clean cap AG, cap analog, that mess up with RNA replication. So after a few rounds of replication, the replication get attenuated. To solve that problem, we came up with clean cap AU. Clean cap AU also has extremely high capping efficiency, about 99%. We talk about making mRNA with cap 1 structure. So far, our focus has been on co-transcriptional capping. So basically, it's one-pot reaction. You need less manufacturing time, and you have less usage of manufacturing space because it's only one pot. You have less reagents, enzyme to validate, and one less step to scale up. However, there is alternative. The alternative is enzymatic capping. So you need 2 bioreactors, one for IVT reaction, one for capping. So in our mind, this is more expensive because you need more manufacturing assets. And it's extra step, and there's some enzymes are maybe challenging to source, and SAM is a methyl donor, has a risk of degradation. How do you cap post-IVT? We've seen this before in cells, right? This is, you can do this in vitro with Vaccinia capping enzyme. There are also 4 steps. First step is to get rid of P. The second step is to add inverted G, and then you add a methyl group. The last step is transfer methyl group to the 2' O position. When you bind the Vaccinia capping enzyme, the protein does work, has 2 domains. One domain does this part, the other domain do the methylation. The thing is, the 2 domains don't always work perfectly in sync. So if one domain work not as well, that would be the limiting step. That's what I heard from my clients, that enzymatic capping is not very reliable. And also, if you want to cap the head of the mRNA, so that head has to be available. Sometimes there's secondary structure that reduce the efficiency of this capping. SAM is this S-adenosylmethionine. It's a methyl donor. You need the methyl donor at this step and also this step. SAM is a large molecule. It's not very stable, has short shelf life, and you also have a BSE, TSE risk. But most important is, in order to do this, you need an additional unit of operation to do the enzymatic capping. Those are some data. Compare CleanCap, enzymatic capping, and ARCA. Those are HPLC mass spec data. So you see, like with CleanCap, you get much capping, much cleaner cap structure compared to enzymatic capping or ARCA. You got more uncapped species here. When you look at the protein expression, CleanCap give you a lot more GFG, a lot more protein expression. To sum it up, when you use CleanCap compared to ARCA, you get much higher yield. It's a single pot reaction, means help you streamline manufacturing. It means time savings because you have less steps, cost savings because there are less enzymes. Most importantly, you get proper capping with cap 1 structure instead of cap 0. We do have a patent on this cap analog, and I'll be happy to talk to you about the commercial license if you are interested. Our CleanCap are used in several COVID vaccines. This one is a BioNTech vaccine, use our CleanCap AG, and this one is a self-amplifying mRNA vaccine from Imperial College of London. It uses our CleanCap AU. We're really proud to help the companies to fight the pandemic. We talk about the cap, we talk about modified uridine. Now let's talk about what else can you do to improve mRNA design. You need a poly-A tail to stabilize mRNA. The standard usually 120 A's. I suspect it came from this paper in 2006 by Dr. Shaheen, Dr. Shaheen's lab. And you can see If you have 128, after 96 hours, you still have lots of transcript left versus at 51 or shorter poly-A tail. So this is in dendritic cells. To get poly-A tail, you can either template it in by PCR or add it to your plasmid, or you can add it by a poly-A polymerase. Most people use templated approach because you get much tighter poly-A distribution. One issue with poly-A tail, if you add it in plasmid, is E. coli as a bacteria does not like poly-A. It has some kind of recombination event to kick out the poly-A if poly-A is too long. In our hand, we can manage to get 88 if it's over 100 or 120. It's not very efficient. However, there are other ways to get around it. It's a segmented poly-A. So basically, you have a section of A, like 40A, and then add a 10 nucleotide of random linker, and then another stretch of A. That helped to get around the recombination event in E. coli. 5' UTRs. When I talk to clients about their mRNA, I heard often people talk about, oh, I know my regulatory sequence, I want to use that. If you want to overexpress your protein, you may not want to use endogenous UTR because we all know proteins are fine-tuned in cells and they may not be designed for mass production. So you probably want to use UTRs of abundant protein. You want a strong Kozak sequence to improve ribosome loading. Secondary structure is questionable. I think the early thinking was you try to avoid secondary structure because it may mess up with ribosome loading. However, that thinking has changed. The other consideration is where is the 5' UTR? If it's the secondary structure, if it's closer to the 5' end, it may not affect ribosome loading. Here's a very good paper discuss 5' UTR, and here's another paper from a group in Stanford. They have very nice discussion about not only 5' UTR but also 3' UTR. The standard UTRs, if you're lazy, you don't want to do anything else, is hemoglobin alpha or beta. The size usually is around 50 nucleotide. 3' UTR, same story here. You may not want to use endogenous UTR. You want to use UTRs of abundant protein to maximize protein production. You want to avoid longer sequence because longer sequence can actually reduce the half-life of mRNA. We all know that microRNA bind to 3' UTR. If you want to improve protein yield, you may want to reduce the predicted binding site of microRNA. Here's a very good paper by BioNTech. They actually did a screen to find 3' UTRs, and the result of this paper is actually in their COVID vaccine. The standard UTRs are, again, hemoglobin, if you don't want to do much with it, hemoglobin alpha or beta. The size usually is around 100 nucleotide. Now we talk about coding sequence. With a coding sequence, you can do codon optimization here. To use more frequently used codons, you can manipulate the secondary structure to maximize protein translation. You can also maximize GC content. There's paper from CureVac a few years ago. They discussed that if you maximize GC content, you may not need to use modified uridine. You can do U depletion. If U is the one give you the problem, you can use a codon with the least number of U's. Here's a real world example. This is BioNTech COVID vaccine is a very elegant molecule. Do they use modified uridine? Yes, they use N1-methyl-pseudo-U. What cap do they use? They use our clean cap with 3-OMe. For 5' UTR, they use human alpha globin with optimized Kozak sequence. They also have a signaling peptide here for ER translocation. For coding sequence, they use the SARS-CoV-2 spike protein with the 2 mutation discovered by an NIH scientist to help the spike protein remain in the pre-fusion conformation. For 3' UTR, this one is from that paper I mentioned before. They discovered by doing screening. For poly A, they use segment poly A with 30A linker and 70A. We talk about mRNA design, and so now how do you make it? You need to start with linearized DNA. It can be either linearized plasmid or PCR product if you don't need a lot of mRNA. And then you put that into an IVT reaction, and then you do buffer exchange, purification, another buffer exchange, and finally QC. If you want to make it yourself, we do have protocols. We also have a method paper, give you the recipe for making mRNA. We also supply catalog mRNAs, so we invite you to compare. For example, you can make a Firefly luciferase mRNA to see how the mRNA you made compare with our catalog product. And then you will know if your process is efficient or not. Or we can make the mRNA for you. We have over 10 years of experience making mRNA. We can help you to shorten mRNA product development time because we have optimized UTRs in our plasmids. We also have process development expertise to tweak the IVT reaction to maximize yield while reducing impurities. We can make mRNA of GMP quality up to phase 1. We also have purification expertise and experience in quality control assay development. We talk about how complex the mRNA design is, and this is what we can help you. Because we have a plasmid with optimized UTRs, we take care of capping tail, 5' UTR, 3' UTR, so we allow you to focus on your own molecule, your own CDS. So that can help you to expedite your product development. We can help you to manufacture mRNA from RUO grade all the way to gram scale GMP grade material. We also provide analytical method development support and also as well as stability studies. mRNA is very unique in a way. First of all, it's safe because it's RNA, there's no genome integration. It's fast because it gets translated into protein right away, bypass nuclease. And this is important for non-dividing cells because for non-dividing cells, it's very hard to get DNA into those nucleases. However, mRNA is short-lived. It can be a negative thing that in other applications may not be as critical. Above it all, it's very easy to manufacture compared to antibody, which need to be produced in CHO cells, or AAV, which need to be packaged in HEC293 cells or other producer cells. There's no complex structure. mRNA is a very large molecule, but compared to protein, it's a lot simpler. The challenge of mRNA is really in the purification, especially double-strand RNA purification. The biggest application for mRNA now is vaccine. We all know that. All the three features of mRNA, clean, fast, short-lived, are all good for vaccine because you just need to present the antigen to the immune system. You actually don't want those antigens to last too long. We all know how fast you can bring an mRNA vaccine to market. The traditional development time for vaccine is about 10 years. And with COVID vaccine, both Moderna and BioNTech brought it to market in like less than a year. A good example is a COVID vaccine, but people also work on HIV and influenza. This is second biggest application for mRNA is gene editing. Why? Because all those effector protein for gene editing, either Cas9 or zinc finger or transposase, are nasty proteins because they cut. They cut DNA in your nuclease. You don't want those proteins to hang around too long. So rather than delivering them in plasmid or a lentivirus, you want mRNA to minimize off-target effect. Because there are mRNA, they're safe, there's no genome integration, and also compared to protein, you can deliver mRNA in an LNP. It's hard to deliver, for example, Cas9 protein in vivo. How it works, you have your LNP get into cells, mRNA, Cas9 mRNA get translated into protein, complex with guide RNA into nuclease, and do your gene editing to edit genes. And we all know Sangamo is a company who has a product based on zinc finger, and zinc finger is delivered as mRNA. Immuno-oncology is another big application for mRNA. You can either use mRNA to add antigen or antigen receptors, or you can use Cas9 mRNA with sgRNA to edit out HLA to make cell therapy allogeneic. One good example is to present antigen. Here, one example is a Duke PP65 LAMP vaccine against glioblastoma. So you introduce the PP65 antigen into cells and then present the cells to immune system to elicit immune response against glioblastoma cancer cells. And here is to present antigen receptor. Example is Cartesian has mRNA-based CAR-T cell therapy against the BCMA. So basically, instead of using lentivirus to introduce a CAR, they use mRNA to introduce a CAR here. And this quadrant is really talking about adjuvant function of double-strand RNA. So we know double-strand RNA elicit immune response, but for vaccine, it can be a good thing to help to prime the immune system to make the immune response stronger. And here is to use the mRNA to make antibody. That's how we use mRNA to make antibody. We all know if you make mRNA in CHO cells, it's complex. So people thought rather than using CHO to make antibody, we can just introduce light chain and heavy chain into cells and let the cells make its own antibody. So basically you have expression within hours. You do need repeated dosing, but it's cell-free production, right? However, we all know mRNA is short-lived, so you probably need repeated dosing. And here you also need IV administration. So there are not a whole lot of talk about using mRNA making antibody now. However, I heard, like, for example, bispecific antibody is really difficult, to make. So maybe mRNA has a place here. And also notice the dosage. So this is one of the clinical trials using mRNA encoding chikungunya virus antibody. If you talk about vaccine, you're talking about 30 microgram. For BioNTech vaccine, 100 microgram for Moderna vaccine. Here you're talking about 0.3 milligram per kilogram. So the quantity of mRNA is very high. There are other applications, for example, protein replacement or rare disease. TriLink Bio has a clinical program treating cystic fibrosis. So this is a disease with lots of mutations. So rather than correct a mutation one by one, the thinking is you should just deliver a functional protein into the patient. So they have CFTR mRNA and delivered through nebulizer to the lung to treat the disease. Another example is glycogen storage disease 1 alpha. Ultragenics has a program, and I think they just obtained rare disease, orphan drug status for their drug. And people also use mRNA for cardiovascular disease, and very early on, one big application for mRNA was actually iPS reprogramming and differentiation, because mRNA is super clean compared to plasmid or virus. This is our beautiful facility in San Diego. We have 118,000 square feet and 15 clean rooms, either ISO class 7 or ISO class 8 to make GMP grade mRNA. I'll be happy to answer questions, and thank you for your attention.
Thanks, May, for that fantastic presentation and for kicking off our mRNA application series. The audience has been actively submitting questions, so let's get to them. Please feel free to continue to submit your questions. Our first question: why would a manufacturer of mRNA-based vaccines want to outsource part of their production process to a CDMO?
Why would they want to? So I think maybe they're not ready, so they're like large companies. We all know they know how to make monoclonal antibody in CHO cells, right? mRNA is still relatively new. I remember when Moderna went public about 3 years ago, we're all talking about, give it 10 years, maybe, Moderna is ready for a prime stage, but then COVID accelerated everything. So basically, for us, for CDMO, we already have the infrastructure put in place. We can make mRNA up to phase 1. So if you really want to accelerate your product development right now, believe it or not, many people are still making COVID vaccine. So COVID window is this much, right? If you want to get it in a window, maybe rather than developing your own expertise, you'll contract to CDMO who know how to do it.
Thanks, May. So a couple questions regarding mRNA manufacturing. Can TriLink help me design my mRNA?
We talk about, we have our proprietary, we call it proprietary plasmid. We allow people to use it. You just have to sign a MTA. So we designed the 5' UTR, 3' UTR. We have poly-A. We have, of course, we have PAP. What we don't do is we don't really help people with your own coding sequence, right? So that we leave it to you. That's your own science. We take care of ancillary properties of mRNA.
Great, thank you. Along the lines of mRNA manufacturing, which base might be better, pseudouridine or N1-methylpseudouridine?
So in our hand, when we make mRNA ourselves, we found N1-methylpseudouridine performs better than pseudouridine.
Thank you. Do you provide LNP packing service?
That's a very good question, because I was just talking to an LNP provider yesterday. So we thought, okay, mRNA, like I said, manufacturing actually is not that hard. The hard part is really LNP. So we don't provide LNP packaging, but we do work with a few fill and finish companies. And if you reach out to me after this, I'll be happy to give you the names of those companies who can do LNP packaging for you.
Thank you. A couple of CleanCap questions. So if someone's using ARCA right now, how can they switch to CleanCap?
Yeah, so ARCA, and also if you use enzymatic capping, it started with G. Why it started with G? Because remember, T7 can incorporate G, it cannot incorporate A. So if you want to switch to CleanCap, you need to do some kind of mutagenesis work. So you can actually swap out the G with A, and then you can proceed with CleanCap. But for you to use CleanCap after the TATA, after the T7 promoter, right? There are 20 nucleotide, the first base has to be A for you to use CleanCap, AG or EU.
Thanks, May. Before we move on to the couple other CleanCap questions, let's shift back to just LNP one more time. Are there other commercial transfection reagents that you might recommend for mRNA delivery in vitro to cells or in vivo?
For in vivo, I think LNP is probably the best way, because we borrow all this from siRNA, right? You know, siRNA made it successful. For in vitro, for cell experiment, mRNA is large. When I talk to customers, you can either do electroporation, or maybe you try the lipofectamine.
Thanks, May. As far as CleanCap goes, do you offer GMP grade?
Yes. So is really GMP. We call it GMP for further processing, because it's not API. It's not an active pharmaceutical ingredient, right? For the ARCA version, for the AG with 3O ME, that product, we should be able to deliver GMP grade. And then, we do have a path to make GMP grade AG and AU, but that will be later.
Thank you. Do I need a license to use CleanCap?
Yes, we do give non-exclusive commercial license to manufacturers upon commercialization. So if you just do clinical trials or research, you don't need a license. But once you start profiting from the CleanCap, from your mRNA, you need to get a license.
Thanks, May. Along those lines, there's a question asking if you can comment a little bit on the technology transfer process and what happens after phase 1.
So we have, so basically after phase 1, we do, when we make mRNA in-house, we first, because construct is different, right? You may have different GC content, different sequence, right? So first we would do a feasibility study, and then we would do process development. And then we scale up to GMP manufacturing. And so once we made the GMP manufacturing, we have all the documentation for this process, and then we transfer that to you. So our team, we have tech transfer team, and that team will work with the client team to transfer the process, the QC, and everything associated with production to the customer or customer selected CDMO for phase 2, 3, and up.
Thank you. And can our products be used in EU countries for clinical trials?
Say it again?
Can our products be used in European countries for clinical trials?
So I think that one you need to work with your own regulatory agency. So we work with lots of customers, but most of them under confidentiality agreement. We do have a few public cases, right? One is Imperial College of London. Imperial College of London actually made self-amplifying RNA for COVID. We made that construct for them, the phase 1 clinical trial. So that's what I can talk about because it's public information.
Thank you. I believe we might have time for one more. Would it be recommended to try promoters other than T7?
We have the most experience with T7, so I think I know there are other people use SP6, but I heard SP6 versus enzyme is a lot more expensive than T7. And then I think it's just like how you move the field forward, right? If there are lots of people working on it, you have more improvement. So for us, actually, when we make the IVT, we just use wild type T7. Then there are all different flavors and modifications of T7. So basically, again, our experience is largely T7. We can't really tell you a whole lot if you use SP6.
Thank you, May. That concludes our question and answer session. Thank you for taking the time to join us and for your questions. I'd also like to thank our speaker, May Guo. As a reminder, the webinar will be archived on the TriLink website, and we'll send you the link within a couple of days. Have a wonderful rest of your day, everyone.
Thank you.